Hi, I'm Matt Fainiff, President and Chief Technology Officer at Biosurfaces Incorporated. I will be serving as the moderator for today's webcast. Biosurfaces' proprietary electrospinning technology is currently being used for many different applications, ranging from medical devices to diagnostic applications. Our first two webcasts of 2020 focused on the medical device side. Truth be told, this is where we have been primarily focused until I received an email from an investigator from the National Center for Advancing Translational Science, or NCATS, at the National Institutes of Health, asking about using our materials for this application. It really wasn't an area that we had planned to dive into at that point. In today's webcast, we're going to discuss how our Bio-Spun materials are being used as a scaffold to grow cells outside of the body using a Transwell permeable support plate. This provides scientists an important tool in their toolbox because it allows scientists to evaluate various interactions, such as how one cell may interact with another cell, how a drug or biologic agent may affect a select type of cell, or how cells may interact with a material before it is implanted into the body in the form of a medical device. These materials can also provide the three-dimensional structure that currently flat plastic tissue culture plates lack in order to grow specific cell types and tissues such as skin, airway, intestinal, etc. These models can then be used to evaluate specific cell-to-cell interactions, how drug affect these tissues, or to learn about pathways for outside interactions, such as how, for example, coronavirus attacks nasal or airway tissue. As you can see, this simple tool has a lot of potential uses. We've assembled a group of people that are responsible for bringing this vision to reality. During this webcast, we will provide you an overall picture of what a Transwell permeable support is, why this research tool is useful, what applications are possible, why there is a need for a 3D scaffold like our Bio-Spun material, how collaborators are using our Bio-Spun materials, and the future of the technology. I would like to introduce our panelists for this discussion. Dr. Lynsey is a segment director of Cell Solutions at Corning Life Sciences. Lynsey received her PhD in biochemistry, a fellow biochemist, from the University of Nottingham, and her executive MBA from Cranfield University. Lynsey's current responsibilities focus on managing Corning's Cell Solutions portfolio of products. Ms. Rayanne Kassab is a research and development engineer here at Biosurfaces. Rayanne received her Bachelor's of Science degree in bioengineering at the University of Massachusetts, North Dartmouth. Rayanne has been involved with developing medical devices at Biosurfaces for over the past almost coming up on 3 years, and has been primarily the primary developer of the Bio-Spun materials used for this application. Dr. Patrick Hayden is the Vice President of Innovative Research here at Biosurfaces. Pat received his Bachelor's degree of Science in Chemistry from the University of Maryland, a PhD in Chemical Biology from Clarkson University, and conducted postdoctoral studies at the National Institutes of Environmental Health Sciences. Pat has been involved in the fields of tissue engineering and cellular and molecular toxicology research using both in vivo and in vitro model systems for more than 30 years. So let's get started with the conversation with Lynsey. For Biosurfaces, a key area was to find a group that was producing the plastic part of the Transwell permeable supports. And that led us to Lynsey after having some conversations with some people at Corning that brought us to Lynsey. We were excited about the possibility of working with a leader in the life sciences space. And Lynsey, can you give us an overview of what Corning Life Sciences mission is?
Yes, thank you, Matt. Corning Life Sciences is a global leading manufacturer of products to support research for several application areas, including core cell culture, microbiology, bioprocess, cancer research, primary and stem cells, drug screening, disease modeling, lab automation, and more. Our mission is to enable our customers to harness the power of cells to accelerate science and ultimately to help understand, treat, and prevent disease.
And when we first met, Lynsey, I noticed that you wore many hats at your position. Can you provide just our audience a high-level overview of what your job entails?
Yes, so as you already mentioned, my role at Corning is Segment Director for Cell Solutions. I manage the business areas of core cell culture, media, drug discovery, and advanced cell culture. And my team and I are responsible for setting and executing our business strategy. My job is a mixture of working with external parties, for example, customers and business partners, and several internal functions such as manufacturing, marketing, and finance to meet customer needs now and in the future.
That's excellent. And can you tell our audience a little bit about what a Transwell permeable support is and what it's used for?
Yes, Corning Transwell and Falcon permeable supports, or cell inserts, are devices for the compartmentalized study of cells. At the bottom of each insert is a thin microporous membrane. And inserts are available with a variety of pore sizes, membrane types, coatings, and configurations, so a diversity of experiments can be designed. Permeable supports can be used to create many cell models across a wide range of research areas, ultimately to produce a cell culture environment that closely resembles an in vivo state.
And, Lynsey, you showed an immediate interest in our Bio-Spun technology in our first meeting, which was very exciting to our group in general. We always like to see that type of interest in our core technology. What were your thoughts about having a 3D nanofibrous scaffold for cells to grow on?
Well, Corning Life Sciences, and in particular our advanced cell culture team, is focused on 3D cell culture. We have a broad portfolio of products that support organoid, spheroid, and tissue models. Solutions include spheroid microplates, Transwell and Falcon permeable supports, as we've discussed, and Corning Gel Matrix. In addition to 3D products, we also support our customers with technical expertise, helping to ensure they have the education they need to successfully support their research. We are always interested to see what other products could complement the Corning breadth of line. And the Bio-Spun technology stood out for us as a product that could enable different techniques for our customers to work with 3D cell culture and potentially new applications.
And where do you envision the 3D bioprinting and tissue engineering markets that rely on developing this type of technology, say over the next 5 to 10 years?
Well, there's a lot of customer interest in these types of applications, and I can envision this really deepening over the next few years. The tools being used enable a movement towards more in vivo-like models with levels of complexity that are more predictive of disease state and drug responses. I can also see an increased movement towards personalized medicine.
Thank you very much. That's great information. A key part of to this program is developing the various electrospun materials that would be used as membranes for these devices. As I mentioned, the overall program has been ongoing for almost 5 years before Rayanne joined our team at Biosurfaces. However, when Rayanne joined in 2018, she almost immediately began working on producing various Bio-Spun material membranes. Rayanne, how has this program changed since you first started working on it?
Well, when I first started on this program, it was in its very early stages. We would get an individual order from the NIH every now and then, make the requested materials, and use their suggested parameters and send them the order. We weren't even making plates back then. I would have to manually cut circular segments from the sheet that I spun, and that's what we would send them. But now it's a much more complete process. We started making our electrospun materials and began mounting them onto 96 and 24 Transwell insert plates. We expanded this process and really developed a complete method from start to finish, and which made it a lot easier for me. And it's very important because our list of customers is continuing to grow inside the NIH as well as outside, which is very exciting. Another way it changed is that we weren't really doing cell culture when the program first began in-house, and now we are well into developing different cell culture applications, which is where Pat comes in, and I'm sure he will talk about that later. It just went from being something that I would work on in my spare time every now and then to becoming one of my more important projects that involves different people working together in the company.
And so what have some of the challenges been in making these materials for the various applications?
Well, to me, the most exciting times are also the most challenging. When we started working with a new customer, they tell us what they're looking for, what they want to accomplish, and what they hope to get from our product. So it's very exciting to go back and forth with the customer on coming up with various ideas that we want to try in order to give the customer exactly what they asked for. This excitement lasts up until we have tested all the proposed ideas and something is still not meeting what the customer is looking for. So then the trial and error process begins where it can get a little challenging. It's just going back and forth, doing the same process multiple times while changing one little thing or altering one little parameter in order to get to the source of the problem and fixing it. And so that can get a little repetitive and a little challenging, but once we fix the problem, it's all better.
Excellent. And what has been sort of the most rewarding part of what you've been working on in this program?
Well, I would say there are two things that I find very rewarding. First of all, overcoming the challenges that I just mentioned, going through these 10 tries of doing the same thing, but then the 11th time we do it, it's like, okay, this works. So that feels pretty good. But second, having that first meeting or that first call or even email with the customer after we've sent them our product or technology information, and just hearing their excitement about the possibilities or the results once they have tested it. So this all makes it worth it the next time we're going through all the challenging process. And we have in mind that at the end we will have a happy customer, which is kind of the push that we need to get through it.
Yeah, and I can't agree more. I think working with the customers and hearing what they're trying to solve for their problem is really a rewarding aspect to what we do. It's what really drove us to bring this technology forward was hearing a customer have a problem, and we provided a solution, and that will continue on in this project. What do you see as future potential applications for this technology?
I think it could be used for many different things. Our material can be biodegradable or non-degradable depending on what the customer needs. So that opens it up to many different applications, whether it's used for growing different kinds of tissues to predict how it will react to different drugs or how different cells interact with each other. I think the fact that the cells enjoy growing on our materials or our membranes makes it a very important diagnostic tool.
Great, thanks for that information. And while Biosurfaces looks to expand development of various Transwell membrane support plates combined with our Bio-Spun materials, we're also working hard to develop applications using these materials in order to best serve our future customers. For us, being able to provide them various applications is really an important aspect to provide full support to the customer, from the material that we're providing to potential applications that they're going to use. And so this is where Pat really plays an important role. Pat, can you talk a little bit about your interest in the tissue culture models in general?
Yeah, thanks, Matt. Sure. So my interest in tissue culture models dates all the way back to my days as a graduate student. And at that time, our lab was using both in vivo and in vitro models to study mechanisms of chemical toxicity. And then as a postdoc at NIEHS, I continued to use monolayer keratinocyte cultures for toxicology studies. And now these cultures were a single layer of cells cultured on a plastic dish submerged in culture medium. And while they displayed some of the biochemical properties of skin cells, they didn't have any of the structure or functional physical properties of actual skin. I had my first exposure to three-dimensional organotypic skin models when I took my first job with Gillette. These models were cultured on Transwell inserts at the air-liquid interface. The tissues had multiple layers, including a functional stratum corneum with in vivo-like barrier properties. They actually look very much like a real piece of skin, and you can actually apply real products to them, like creams and lotions. Things that you could never imagine doing with a submerged monolayer culture. So I was really intrigued by these models, and I immediately knew that I wanted to keep working with them. One of the skin models that I had been using at Gillette was produced by MatTech Corporation, and I was eventually recruited to join the company. So at MatTech, my interest changed from primarily using the in vitro models as a toxicologist to primarily developing the models in related toxicology applications. And I ended up spending the next 20 years at MatTech producing various 3D tissue models and related applications as commercial products. These models all involve the use of Transwell systems and include various skin, lung, and other types of tissue models. And now my current work here at Biosurfaces involves adapting a variety of open source 3D tissue models to Biosurfaces' novel electrospun scaffold materials.
Excellent, Ben. And why are these types of models important to develop for a customer or in general for general use?
Well, in vitro cell cultures are important fundamental tools in biological research, including toxicology and drug development research. But as we've already mentioned, the submerged monolayer cultures grown on plastic dishes don't reproduce the true 3D structure, behavior, and function that the cells normally have in the body. So the big push now is to develop more types of 3D organotypic tissue models that are better replications of the true structure, behavior, and function that the cells normally have in the body. and this often involves some sort of support or 3D scaffold material. There's also a very big push now to use human cells whenever possible as well, because results obtained from animal cells very often do not translate well to humans. And the ultimate aim and expectation is that these tissue models will lead to more accurate assessment of toxicity tests and faster and less expensive development of therapeutic treatments for human diseases.
And, Pat, I mentioned earlier in the introduction that, Biosurface has been primarily focused on medical devices, and that really, those types of medical implants really rely on understanding how the body is going to interact with an implantable device. So if from an outside perspective, why is it important for biosurfaces to better understand the interactions of cells with our Bio-Spun materials?
Well, one of the most important attributes of the biosurfaces, Bio-Spun materials, is the thickness of the electrospun fibers, which are very similar to the native extracellular matrix fibers. This more natural environment allows cell attachment without elicitation of fibrotic or inflammatory responses, as often occurs with other types of materials. However, beyond a range of potential fiber thicknesses, we also have a broad range of materials with different chemical compositions and surface properties. So a key to developing useful organotypic tissue models from these materials is an understanding of how these various types of cells will interact with the materials so that the optimal material and culture conditions can be developed for the desired application.
And what types of applications, Pat, are you currently working on? And what else do you hope to apply this technology to?
Well, I'm currently working on adapting several epithelial tissue models to Transwell inserts that incorporate Bio-Spun materials. The detailed open source recipes for producing many types of epithelial models are widely available in the literature and are routinely produced in numerous labs all over the world. However, we believe that many of these open source models may be improved by adapting them to Bio-Spun scaffolds. At the moment, my main focus is on development of alveolar air-blood barrier models consisting of human alveolar epithelial cells and endothelial cells. This work is being supported by the NCATs at NIH. Another model I'm currently working on is of the nasal mucosal epithelium. The impetus for development of both of these respiratory system models is for COVID-19 research and related toxicology and drug development applications. I also expect that other types of epithelial models could benefit by adaptation to electrospun scaffolds, including the skin, gut, ocular, kidney, as well as other non-epithelial models such as the blood-brain barrier, neuronal models, liver, tumor models, and so on and so on. The sky really is the limit to what I think we could apply these models, these scaffolds to.
That's excellent, Pat. Thank you so much. And so before we close, I would like to ask each of the panelists their personal thoughts about the opportunities for this technology in terms of what is appealing to them in their respective space. So Lynsey, we'll start with you.
Yes, well, we are pleased to work with Biosurfaces to enable customers to really leverage the proven Transwell permeable supports platform from Corning and combine it with Bio-Spun nanomaterial membranes to further enable complex 3D cellular and tissue biofabrication. It's really exciting to see how 3D cell culture applications are evolving.
Excellent, thank you. And Rayanne?
From my perspective, a lot of the time I'm making the same kind of materials, but maybe tweaking it a little bit to fit what the customer wants. And that's what I love about our process. We can adjust it based on the customer's needs. So all they would have to do is give us their specifications in terms of thickness, surface morphology. In other words, if they want it to be fibrous or film-like or any other specification that they have, and we will take these things into account when we're fulfilling their order, and we would give them what they want and what they have asked for.
Excellent. And last but not least, Pat?
Thanks, Pat. So what's most appealing to me is that the work that we're doing developing these new tissue models is really at the cutting edge of biological science. And this work has the potential to make a real impact in areas such as toxicology and drug development that contribute to improving people's lives.
Thank you, Pat. And I'd like to thank all of our panelists today for taking the time to speak with us. And would also like to thank you for taking time out of your day to listen to our webcast. If you're interested in learning more about our Bio-Spun technology combined with Transwell permeable support plates, which will be hopefully you'll be seeing more of on our website in the next coming months, or would like to make a suggestion about future webcast topics, please feel free to reach out to us at info@biosurfaces.us. To learn more about Biosurfaces, the company, please visit our website at www.biosurfaces.us. We'd love to hear from you. And until next time, thank you so much.